Okay, in this video I'm going to see about the historical background of the cell and the structure and function of subcellular units. Now, according to the cell theory, all organisms are composed of cells, and the unit which can be called living is the cell, which may form a single-celled organism like Chlamydomonas, which you can see the picture on the right, which is a unicellular chlorophyte, or yeast, which you can see the diagram here again, which is a unicellular fungus. So it can be a multicellular plant or animal also. All are made up of cells. Either they are unicellular or they are multicellular. So the cell is the basic structural and functional unit of life. And the level of organization of matter represented by a cell shows that all the characteristics of life. So a single cell, even organisms which are only unicellular, they can perform they show all the characteristics of life. So any stage below the level of the cell cannot be considered living. Robert Hooke examined a cork using simple microscope and gave the term cell to describe the basic units. While Anton Leeuwenhoek was the first to describe and record living single-celled organisms, euglena and the bacteria. On the left side you see the structure of a euglena. This is a protist, and it has a well-organized nucleus here containing the genetic material. They have the chloroplast, and it doesn't have a cell wall, but it has what is called a pellicle. Now here these protists, they are eukaryotes. Although it is a single cell, it can perform all the characteristics of a living organism. Whereas on the right you find the bacterial cell, where the DNA is just found in the center. There is no nucleus, and the region occupied by the DNA is called a nucleoid. There are, as usual, ribosomes, cell membrane, the cell wall, capsule, pili and flagella. Capsule, pili and flagella you get only in some bacteria. You don't get it in all the bacteria. Here is a picture of the cork cells, which was recorded by Robert Hooke. Now in Hooke's drawing, the cells look empty. They look like empty chambers because he was looking at the dead plant matter. Okay, so he didn't look at the living cells. The living cells have a lot of components in them, whereas here these cells are empty because they are dead. And so today we know that living cells are made up of many structures, which we will see in detail. Schleiden, Schwann and Virchow presented the cell theory. According to them, all organisms are composed of one or more cells. The basic structural and functional unit of organisms is the cell, and all cells arise from pre-existing cells. So Matthias, who worked on plants, a botanist, he said that all plants are made up of cells, while Schwann said that all animals are made up of cells, and Rudolf Virchow, he said that all cells came from pre-existing cells, and together they made the cell theory. Organization of cells. There are two kinds of cellular organization, prokaryotic and eukaryotic. So the major difference between prokaryotic and eukaryotic cells is the location of the DNA. In eukaryotic cells, DNA is located in the nucleus, which is a double membrane organelle, while in prokaryotes, the DNA is in the region not membrane enclosed. It is the nucleoid. So eukaryotic means true nucleus, while prokaryotic means before the nucleus. So there are membrane bound organelles which are found in the eukaryotes. And eukaryotic cells are larger than 10 to 100 μm in diameter, while bacteria are about 1 to 5 μm in diameter. So although there are two kinds of cellular organizations which are prokaryotic and eukaryotic, all cells they share the same basic features. That is, they are all bound by plasma membrane, which is a selective barrier. Within the cell, they have a semi-fluid jelly-like substance which is the cytosol, uh, and subcellular components are suspended within the cytosol. The subcellular components, they can be the different organelles, and they carry DNA as genetic materials, and ribosomes are found in all cells. Now, difference between prokaryotic and eukaryotic cells. Here you can, there are diagrams which are given here, which is a bacterial cell and an animal cell which is a eukaryotic cell. Now, if you look at the tabular column, you will find that the organisms in the prokaryotes are the bacteria and archaea, while eukaryotes are the protists, fungi, plants and animals. And I told you about the cell size, where prokaryotes are 1 to 5 μm and eukaryotes 10 to 100 μm in diameter. And prokaryotes, there are many unicellular, where eukaryotes they are multicellular, except the protists and some fungi which are unicellular. Okay, now prokaryotes they originate about 3.5 billion years ago, while eukaryotes their origin is about 1.8 billion years ago, and they have evolved from the prokaryotes. In prokaryotes, you get the cell division, which is just the binary fission. They just divide. Okay, so there's no mitosis or meiosis taking place. Whereas in eukaryotes, you get mitosis, meiosis, or both taking place. Okay, then in the genetic material is the DNA, the circular DNA, which lies free in the cytoplasm. So this region is called the nucleate, as I told you earlier, in the prokaryotes. And the DNA is naked and not associated with proteins. Here it is not associated with proteins, whereas in the eukaryotic cell you find DNA is linear and contained in a nucleus, and it is associated with the proteins. And there are types of ribosomes, 70S and 80S ribosomes. In prokaryotes you only get the 70S ribosomes. Whereas in eukaryotes you get 70S ribosomes in the mitochondria and the chloroplast, and 80S ribosomes are present, may be present and attached to the endoplasmic. Now in the prokaryotes you get few organelles, none are surrounded by a membrane, and internal membranes are scarce, and if present associated with respiration, as the mesosomes you find, of photosynthetic lamellae, and they might be involved in nitrogen fixation also. Now in eukaryotes you get many organelles, membrane bound organelles are present, great diversity of organelles also. So you get nucleus, mitochondria, chloroplast bound by two membranes, while lysosomes, vacuole are bounded by a single membrane. Now cell walls consist of peptidoglycan in bacteria and cyanobacteria, and polysaccharide and protein present in the archaea bacteria. While in eukaryotes, cell walls of green plants and fungi are rigid and contain polysaccharides. Okay, so cellulose in plant cells and chitin in fungal cells. Okay, so you don't get a cell wall in animal cells as such. Then you get the flagella, simple, they don't have microtubules, so they lack microtubules, and they are not enclosed by a cell surface membrane. Okay. While here it is the flagella, they are complex with 9+2 arrangement. We will talk about all that later of microtubules. And respiration in the prokaryotes you get mostly by the mesosomes, and in the eukaryotes the mitochondria help in the respiration. In photosynthesis, no chloroplast, they have photosynthetic lamellae, while here in the eukaryotes you get chloroplasts, they contain membranes which are stacked into lamellae or grana. And nitrogen fixation you find in the prokaryotes, where eukaryotes you don't have that nitrogen fixing ability. When you look at the animal cell, you'll find that the plasma membrane enclosing the cell, ribosomes which are free within the cytosol, and even some bound to the rough endoplasmic reticulum and the nuclear envelope. And you find the Golgi apparatus, organelle active in synthesis, modification, sorting and secretion of the cell products, and lysosome, digestive organelle where macromolecules are hydrolyzed. You get the mitochondria, organelle where cellular respiration occurs and most ATP is generated, and peroxisome, organelle with various specialized metabolic functions, produce hydrogen peroxide as a byproduct and then converts it to water. Microvilli, projections that increase the cell surface area. Microvilli you find in cells lining the gut to increase the surface area for absorption. Then you find the microtubules, intermediate filaments and microfilaments which form the cytoskeleton of the cell. All this forms the cytoskeleton and reinforces the cell shape and the functions and cell movement, all that. And then the centrosome, region where the cells microtubules are initiated. So you get the centrosome, and contains a pair of centrioles which are involved in the cell division, and flagellum, motility structure present in some animal cells only. You get this flagellum which is found in some animal cells and formed of microtubules, and an extension of the plasma membrane. And you have the endoplasmic reticulum, it's a network of membranous sacs and tubes, and active in membrane synthesis and other synthetic and metabolic processes. So rough and smooth regions are present here, and we'll see about all that in detail. And then you have the nucleus. The nucleus has been cut open to show you the nuclear envelope, which is a double membrane enclosing the nucleus, and it is perforated by pores. It is continuous with the ER, endoplasmic reticulum. There's a nucleole is also present, involved in the production of ribosomes, and there may be one or, depending on the type of cell, there can be one or more nucleoli. And chromatin material consisting of DNA and proteins. So it is visible in a dividing cell as individual condensed chromosomes. Or we will be looking at all these cellular organelles more in detail. This diagram structure of the animal cell is from your resource book. You can look at that there. Now here is a structure of a plant cell. You find here also you have the cell wall. You can see the cell wall at the bottom of the diagram. You get the cell wall, outer layer that maintains the cell shape and protects the cell from mechanical damage. So in animal cell you didn't get the cell wall. So you have a cell wall which gives protection, and it's made up of cellulose and other polysaccharides and protein. And you can see the wall of the adjacent cell also, which has been cut off here. Now in the cell wall you find some openings here which are called the plasmodesmata. So there you can see the plasmodesmata, cytoplasmic channels through cell walls that connect the cytoplasm of adjacent cells. Adjacent means these cells, nearby cells, they are connecting the adjacent cells. So they are cytoplasmic connections between adjacent cells. Okay. Then inner to that you get the plasma membrane which has been marked here. That is common to all the cells. Then going up further you find the peroxisomes, and mitochondria. We've seen that in the plant cells. And here you get the chloroplasts. So they are the photosynthetic organelles. They convert energy of sunlight to chemical energy and store it in sugar molecules. And then you find the cytoskeleton, the microfilaments and the micro tubules. They form part of the cytoskeleton, and you get a central vacuole, a very large central vacuole which you don't get in the animal cells, a prominent organelle in older plant cells. The functions include storage, breakdown of waste products, and hydrolysis of macromolecules. Enlargement of the vacuole in a major mechanism of plant growth. Okay. Then you find the ribosomes there, ribosomes and smooth endoplasmic reticulum, rough endoplasmic reticulum. We call that rough endoplasmic because of the presence of the ribosomes on the ER. And then you have the nucleus again, which has been cut open here with a nuclear envelope, nucleole as chromatin. And then you find the Golgi apparatus. And this is the structure of the plant cell, which you can see in your resource book. The plasma membrane, we'll talk about that in detail. So it is the outer layer of the cytoplasm, and all cellular membranes resemble the ultrastructure of the plasma membrane. So Singer and Nicholson put forward the fluid mosaic model of the cell membrane, which we are also following. Now this plasma membrane is made up of phospholipids, which are the most abundant type of lipids in the plasma membrane, and protein. It's about 7 nm thick and made up of a phospholipid bilayer, which are amphipathic. That means they have hydrophilic and hydrophobic parts, hydrophilic heads and hydrophobic tails. Now the hydrophilic heads of the phospholipids, they face outwards into the aqueous environment of the inside and the outside of the cell, and the hydrophobic hydrocarbon tails, they face inwards and create a hydrophobic interior. Now here you find the phospholipids, which are the amphipathic molecules. So it's a bilayer held by weak hydrophobic interactions between the tails, you can see that, and hydrophilic and hydrophobic layers restrict the passage of any substances, of many substances. So individual phospholipids, they can move within the bilayer, allowing for membrane fluidity and flexibility. And the fluidity allows for the breaking and reforming of the membranes also. Now, according to the fluid mosaic model, since phospholipid molecules are movable, they provide the fluid nature of the membrane, while protein molecules embedded randomly contribute to its mosaic nature. Some of the protein molecules penetrate all the way through the membrane and are called transmembrane proteins. Some penetrate only part of the way into the membrane, are called integral proteins. Most of the integral proteins are transmembrane proteins, which have hydrophilic channels. So these are act as pores through which the ions and certain polar molecules can pass. Now here is the structure of the fluid mosaic model, showing you the phospholipid bilayer, the phospholipids with the polar heads pointing into the extracellular and extracellular fluid and the cytoplasm, and the hydrophobic interior formed by these fatty acid chains of the phospholipids. You find the proteins which are just dotted, giving a dotted appearance of mosaic appearance, and among the proteins you find the integral proteins which are spanning the membrane, transmembrane proteins. So most of the integral proteins are called the transmembrane proteins, and they have channels for the movement of the hydrophilic substances. Some glycolipids and glycoproteins also are found, which are formed of carbohydrate and protein, or carbohydrate and lipid, which helps in signaling as signaling molecules. Cholesterol also is present to give stability to the membrane. And these proteins, some of the proteins, these peripheral proteins and all that, they just lie on the outside of the membrane and the inside of the membrane, and they are attached by some cytoskeleton elements. Now here you have a diagram of the plasma membrane again. So here you find the phospholipid bilayer. Actually, this particular diagram was taken from Campbell. And you find that I think that actually the plasma membrane, it is a boundary that separates the living cell from its surrounding and controls the traffic into and out of the cell it surrounds and exhibits selective permeability. So I told you lipids and proteins, they are the stable ingredients of the membranes. Carbohydrates also can be found. Now, so we said the phospholipids are the most abundant and they are amphipathic. That means they have both hydrophilic and hydrophobic regions. So the lipids, mostly the lipids and some proteins, they can shift about laterally in the plane of the membrane. So the membrane, they remain fluid as the temperature decreases. Now cholesterol at high temperature makes the membrane less fluid by restraining the phospholipid movement. So the membranes must be fluid to work properly. So the fluidity of the membrane affects its permeability and the ability of the membrane proteins to move to where its function is needed. Now proteins, like a mosaic tile membrane, is a collage of different proteins embedded in fluid matrix of the lipid bilayer. Proteins determine most of the membrane's functions. So different types of cells contain different set of membrane proteins. Now the membrane proteins, integral and peripheral proteins. Integral proteins, they penetrate into the interior, into the hydrophobic interior of the lipid bilayer, and the majority are transmembrane proteins which span the membrane. Other integral proteins extend only partway into the hydrophobic interior, while the peripheral proteins are not embedded in the lipid bilayer at all. They are loosely bound to the surface of the membrane. On cytoplasmic side of the plasma membrane, some membrane proteins are held in place by attachment to the cytoskeleton, and extracellular side by some proteins that are attached to fibers of extracellular matrix. So you find a large extracellular matrix here found on the outside of the cell, the extracellular matrix. Okay, and so the cells can recognize other cells by binding to molecules uh, containing carbohydrates on the extracellular surface of the plasma membrane. Now membrane carbohydrates, they are short branch chains bonded to lipid forming glycolipid. So a steady traffic of small molecules and ions move across the plasma membrane in both directions. Now sugars, amino acids, nutrients can pass into the cell and waste can leave. So the cell takes in oxygen and expels carbohydrate and regulates the ions as sodium, potassium, calcium and chloride. Now non-polar molecules such as hydrocarbons and carbon dioxide and oxygen, are hydrophobic and they dissolve in lipid bilayer and cross easily without the aid of the membrane proteins. Now hydrophobic interior impedes the passage of ions and polar molecules. So they have to pass through the hydrophilic regions. Okay. So specific ions and a variety of polar molecules can't move through the cell membrane on their own. So then hydrophilic substances can avoid contact with the lipid bilayer by passing through transport proteins that span the membrane. Some proteins are not embedded in the lipid bilayer at all and are loosely bound to the inner surface of the membrane called peripheral proteins. We've seen all this in the previous slide. So some proteins and lipids have short branching carbohydrate chains like antennae forming glycoprotein and glycolipids, and animal cell membranes may contain cholesterol molecules randomly integrated in the lipid bilayer. Now cholesterol molecules, they provide flexibility and stability to the membrane. By reducing membrane fluidity at moderate temperatures and prevent membrane solidification at low temperatures. So the two sides of the membrane may differ in composition and function. This is a diagram from the resource book, which you can go through the same thing. Functions of the plasma membrane. It surrounds the cytoplasm of the living cell physically, separating the intracellular components from extracellular environment. The plasma membrane is selectively permeable and regulates the exchange of material for survival. The proteins embedded in the plasma membrane identify the cell, enabling nearby cells to communicate with each other, that is, cell recognition. And some protein molecules act as receptor molecules for interacting with specific biochemical hormones, neurotransmitters, and immune proteins. And some proteins in the cell membrane attach to cytoskeleton fibers and help to maintain the shape of the cell. And some proteins in the membrane act as enzymes. The microvilli on epithelial lining of some parts of the gut, they contain the digestive enzymes in their cell surface. These are functions of the plasma membrane. The proteins, some of the functions of the proteins that are found in the plasma membrane. So you find them, they help in the transporting of substances, they serve as enzymes, they serve in signal transductions as receptors for certain chemical messengers, and cell recognition, and intercellular joining of adjacent cells, and attachment of the cytoskeleton extracellular matrix. So these are some of the ways in which the proteins function. Now coming to the nucleus. I told you it's the most prominent organelle, consists most of the genes, having an average diameter about 5 μm, enclosed by double membrane cover called the nuclear envelope. Now this nuclear envelope is composed of two membranes, an inner and outer membrane, and separated by a space of 20 to 40 nm. Now nuclear envelope is perforated by nuclear pores, which have a pore complex to regulate the entry and exit of substances. Now it has a nuclear lamina made up of protein filaments which line the interior of the nucleus. Here you can see the diagram of a nucleus which has been cut open to show the inside. Now the nucleus contains most of the genes of a eukaryotic cell. Now some genes are located in the mitochondria and the chloroplast. Now this nucleus is the most conspicuous organelle, it's about 5 μm in diameter, and the nuclear envelope encloses the nucleus, separating its contents from the cytoplasm. Now this nucleus is a double membrane organelle, and these two membranes are separated by a space of 20 to 40 nm. And the envelope of the nucleus is perforated by pores about 100 nm in diameter. And these pores, there is a pore complex which is found there, which lines each pore. This pore complex lines each pore, and it regulates the entry and exit of proteins and RNA and large complexes of macromolecules. Now, except at pores, nuclear side of the envelope is lined by a nuclear lamina, which is a net-like array of protein filaments that maintain the shape of the nucleus by mechanically supporting the nuclear envelope. Also, the nuclear matrix, it presents a framework of protein fibers extending throughout the nucleus interior. Now the nuclear lamina and the matrix help organize the material so it functions efficiently. Now within the nucleus, the DNA is organized into discrete units called chromosomes. So that they are structures that carry the genetic information. Now these chromosomes, they contain a long DNA molecule associated with many proteins. These proteins help to coil the DNA. So the chromosomes, they are a complex of DNA and proteins. So the complex of DNA and proteins only make up the chromosomes. And this, when it is in a non-dividing cell, we call it as chromatin because it appears in a diffuse nature. When the cell is not dividing, the stained chromatin appears as a diffuse mass in micrographs. When cells prepare to divide only, the chromosomes, they start to coil. Now in human, there are about 46 chromosomes, while as in the egg and sperm there are 23 chromosomes. Fruit fly contains about 8 chromosomes and 4 in the sex cells. Inside the nucleus you also get nucleoli. Nucleolus is the singular. It is in non-dividing cells, the nucleus contains the nucleolus. So here you find a mass of densely stained granules and fibers. Now rRNA synthesized, ribosomes made from protein and the rRNA. Now the nucleus directs protein synthesis by synthesizing mRNA according to instructions from the DNA. So mRNA is made in the nucleus. rRNA also is made in the nucleus. So once mRNA reaches the cytoplasm, ribosomes translate the mRNA's genetic message into the primary structure of the specific polypeptide. So remember that the DNA found in the nucleus, it has the code on it, and the mRNA brings the information or the codon from the DNA. So the code from the DNA is transcribed onto the mRNA. mRNA brings the code out into the cytoplasm, and where the ribosome is present, and the code is translated into the polypeptide chain together with the tRNA molecule. So here in this diagram you can see a closer view of the nuclear envelope with the complex and the chromatin also. So nuclear matrix made up of protein filaments extended throughout the interior of the nucleus. All that we've seen in the previous slide. Chromatin and nucleolus embedded in the nuclear matrix. Nucleolus appears as darkly stained granules with fibers adjoining part of chromatin. Chromatin appears as diffuse mass in non-dividing cells composed of DNA and proteins. During nuclear division, chromatin condenses, tightly coils and forms the chromosomes. All this we've seen, I spoke to you about in the last slide. Now here again in this picture you find the chromatin, DNA double helix here. It is coiling around the histone proteins to form the nucleosomes, and 11 nm in diameter, and these again coil, again it further coils, the nucleosomes fold accordingly, and then they form 30 nm fibers. The 30 nm fibers, they are thrown into irregular loops to form 300 nm fibers, and these 300 nm fibers again coil to form what are called the chromatids. So in dividing cells only you find the chromatid which is about 700 nm. 700 nm fibers. So each chromatid is a 700 nm fiber, and these chromatids, chromatids, when they duplicate themselves, you find they form chromosomes which are held together at the centromere, and there you find that the length of this is about 700 nm in length of these fibers. The functions of the nucleus is it controls all cellular activities. synthesize DNA to produce new nuclei for cell divisions, synthesize rRNA and ribosomal subunits required for protein synthesis through the nucleolus, synthesize mRNA and tRNA according to the information present on the DNA, store and transport genetic information. Ribosomes are subcellular components which carry out protein synthesis. Now they consist of two subunits, larger subunit and a smaller subunit, and they are composed of rRNA and protein. They are complexes of rRNA and protein cellular components that carry out protein synthesis. Now there are ribosomes, there are two types, 70S and 80S ribosomes. 70S ribosomes are found freely in the cytoplasm of prokaryotes, matrix of mitochondria and stroma of chloroplast. 80S ribosomes are found only in the eukaryotes. So based on the nature of the presence, 80S ribosomes are categorized into two types, free ribosomes and the bound ribosomes. Free ribosomes are freely available as a group in cytoplasm. Bound ribosomes are attached to the membrane surface of the rough endoplasmic reticulum, and their function is in protein synthesis. Now you look at the structure of this ribosome. It has two subunits, a large subunit and a small subunit. Now here you have the ribosomes are involved in the protein synthesis. So it has an mRNA binding site. You see the mRNA in the center with the codon which it has brought out from the nucleus, and you find that both these subunits are assembled here with the mRNA and the tRNA. So the mRNA, the ribosome has an mRNA binding site and 3 tRNA binding sites, which are, so the tRNA binding sites are A, P and E. So A is the amino acyl tRNA, P is the peptidal tRNA and E is the exit. So you find the codon AUG on the mRNA, it is matching with the anticodon UAC on the tRNA, and the tRNA here in the P site is also attached to the amino acid methionine. So protein synthesis is starting at this particular place. Now we come to the endoplasmic reticulum. It is an extensive network of membranes. You can see this extensive network of membrane consists of a network of membranous tubules and sacs called cisternae. So the endoplasmic reticulum membrane separates the internal compartment of the endoplasmic reticulum, lumen or cisternal space, from the cytosol. So this is continuous with the nuclear envelope. The smooth endoplasmic reticulum is the outer surface. The smooth outer endoplasmic reticulum outer surface lacks ribosomes, whereas the rough endoplasmic reticulum is studded with ribosomes on the outer surface of the membrane. Now the smooth endoplasmic reticulum, it synthesizes lipids, metabolism of carbohydrates, detoxification of drugs, and involves the storage of calcium ions. And enzymes of the smooth endoplasmic reticulum are important in the synthesis of lipids, oils, steroids, and membrane phospholipids, and steroid hormones secreted by the adrenal glands. The cells that synthesize and secrete these hormones in the testis and ovary are rich in smooth endoplasmic reticulum. Other enzymes of the smooth endoplasmic reticulum detoxify drugs and poisons, especially in the liver cells. Now, functions of the endoplasmic reticulum. Many cells secrete proteins produced by the ribosome attached to the rough endoplasmic reticulum. Like certain pancreatic cells, they synthesize proteins like insulin in the endoplasmic reticulum, and this is sent this hormone into the blood. So it makes secreting proteins and membrane proteins and phospholipids for its own membrane. So according to this diagram, you can see the nuclear envelope, and attached to it is the cisternae of the rough endoplasmic reticulum with the ribosomes attached and the smooth endoplasmic reticulum present. So the rough endoplasmic reticulum consists of flattened sacs and ribosomes bound to the surface. Proteins synthesized by ribosomes move into the lumen of the ER. The functions: transport proteins synthesized by ribosomes, synthesizing glycoproteins, produce transport vesicles, facilitate the growth of the own membrane by adding phospholipids, proteins and carbohydrates. Therefore, it is a membrane factory. Smooth ER is a network of tubular sacs without ribosomes. Membrane bound enzymes are present. Functions: synthesizes lipids including oil, steroids and phospholipids. Metabolism of carbohydrates. Produce transport vesicles to transport within the cell. Involves in detoxification and stores calcium ions. Now here we see the Golgi apparatus or Golgi complex. This Golgi complex consists of flattened membranous sacs which are called cisternae, and it has a cis or receiving side of the Golgi apparatus and transphase or shipping side of the Golgi apparatus. The cis phase is located near the ER or endoplasmic reticulum, and transport vesicles move material from the ER to the Golgi apparatus. Vesicles that bud from the ER can add its membrane and contents of its lumen to the cis phase by fusing with a Golgi membrane, and the transphase gives rise to vesicles that pinch off and travel to other sides. The products of ER are modified during this transit from cis region to trans region of the Golgi apparatus. Now in this diagram also you can see the vesicles that move from the ER to the Golgi, and the vesicles coalesce to form new cis Golgi cisternae, and cisternal maturation, Golgi cisternae move in a cis to trans direction, and vesicles form and leave Golgi carrying specific products to other locations or to the plasma membrane for secretion, and vesicles transport some proteins backward to less mature Golgi cisternae where they function, and vesicles also transport certain proteins back to ER, their site of function. So again, the Golgi apparatus is a stack of flattened sacs of cisternae. Inner and outer surfaces can be identified as cis and trans phase respectively. Cis phase located near the ER to receive the vesicles from ER. Trans phase gives rise to secretory vesicles which are budded off and travel to the other side. Golgi complex is abundant in secretory cells. Remember, Golgi complex is abundant in secretory cells, and the function of it is collecting, packaging and distribution of materials, manufacturing cellulose and non-cellulose cell wall components such as pectin, and production of lysosomes. Here you have another diagram of the Golgi apparatus, the cis phase and the trans phase. You can note them, how you can see how the cisternae are arranged from the cis to the trans phase, how they mature, and the incoming transport vesicle with the products from the endoplasmic reticulum, and you can see the newly formed vesicles on the trans phase, secretory vesicles which are budding off to go to the respective destination. Here you can, it's a review relationship among organelles of this endomembrane system. So the nuclear envelope is connected to rough endoplasmic reticulum, which is also continuous with the smooth endoplasmic reticulum. Membranes and proteins produced by the ER flow in the form of transport vesicles to the Golgi. Golgi pinches off transport vesicles and other vesicles that give rise to lysosomes, other types of specialized vesicles and vacuoles. Lysosome is available for fusion with another vesicle of digestion. Transport vesicle carries proteins to plasma membrane for secretion, and the plasma membrane expands by fusion of vesicles. Proteins are secreted from the cell.